Hi, Frank. How are you?
Hey there, I'm fine. How are you?
Good, good. Thank you.
I guess you were just doing the introduction to Clubhouse.
For the room on Monday, which will be really with Peter Ruprecht. Will be really interesting. So yeah, I'm looking forward to it. Hi, Jeff. How are you? Oh, no.
Yeah, that used to happen when the small is still. Yeah, the room is still small. The smallest still room. Yeah, and people are just checking in and they want to have this conversation already ongoing, and then they're kind of what's the word? Yeah, yeah. Anyway, happens at the beginning.
Yeah. Hi, Sophia. We'll start on top of the hour. Just Hi, David. How are you today?
Full of expectation. That is what I was looking for. So yeah, full of expectation. You're already expecting an ongoing discussion and want to listen in.
Yeah, I kind of get it. So it's fine. It's kind of boring the first 15 minutes. So, but I shared the paper in the chat. Should be open access. So if people want to look at it, the presentation is also pinned on top of the room. So if if anyone wants to have a head start and look at it, I think both the paper and the presentation are really interesting. So, yep, looking forward to the talk. Our guest speaker will be here in a few minutes, so thanks for coming.
I would just say upfront, I have no idea in this topic, so I won't have any questions. But I'm curious. I'm listening. But that is kind of an area where I have no knowledge or not much.
Yeah, sometimes with the presentation, like something comes up, like a question. So,
I mean, it's
yeah.
related to the whole to the whole immunology with COVID. So I'm kind of that the T cells are there and what they're generally doing. That's something I know, but I have no idea about the cells themselves.
Oh, yeah, I'm not sure if that will be discussed, but oh yeah, there's development of these cells. Yeah, she will explain a few developmental steps and lineage before she goes in. So yeah, that's good. Hi, Kim. How are you?
Hi, Katerina. I'm good. How are you?
Good, good. Thank you for coming. We were starting
Sorry
to look.
again for the last-minute rescheduling. Thank you for accommodating me.
Oh yeah, don't worry. It's informal, so we are happy that you come and take the time to talk with us. So no worries. Thanks for giving us a heads up. And meet Frank. Frank, meet Kim. We were already
Hey there.
we were already looking at the slides a little bit, but we have still around 4 minutes before we start, so
Yeah, no problem. And let me know if my connection sounds okay. I'm in a new building, and I think the signal sometimes is spotty. So do let me know if that becomes a problem.
okay. Yeah, for now you sound perfect. So
Okay.
yeah. And happy Friday. I hope your week wasn't
Happy
too stressful.
Friday. It's a long weekend.
Yes. It's a long weekend, exactly. You having, you're doing something on Labor Day.
Absolutely nothing, in a good way.
That was perfect.
What about you?
Yeah, we're going to the beach for a few days before my kids' school. Uh, for whatever reason, they start earlier than other schools. Like they're in a charter school here in New York City, so they kind of start a few weeks earlier than everyone else. But so I'm kind of happy that we get this long weekend before. It's such a long stretch until they have off again.
That's right. Yeah, really gotta recharge as much as possible, and then have the endurance.
Exactly. So, and their school starts so early. Like, I feel so bad for them. Their
How
school
early? Like
starts at
7, 8?
7:30. It's early, right? I feel
It
bad.
is early. I thought some European countries decided to start later because they think that actually has a better impact on kids' performance later in life. I thought there was some study about it. I was like, yes, should have done it when I was a student.
Yeah, I agree. So yeah, in Portugal, schools start at 9. When I went to school in Germany, it started at 8:15, I believe. So, but 7:30 is way too early.
7:30 is way too early, and the parents, they have to get up much earlier. Oof.
Yes, and then they don't. I mean, for me, it's good, but they still go until 3:30. So it's a very long day for them of sitting,
It
just
is.
sitting. Well, anyhow, I just feel bad for them. I mean, I can, for traffic and everything, it's kind of better for me, around that time, like less
True.
traffic but anyhow.
Yeah, but that is a draining day. How many hours is that? 8 hours? Oof.
Yeah, so
Do they still have after-school activities like clubs or
um yeah, so I tried to keep it fairly low, so they have
sports?
2 things. Yeah, they have like 2 sports. They used to have more, but it was just too stressful. So this year we might just stay with 1 or 2, at least in the beginning. Um, yeah, I think you know. Yeah, it's 2:31, and the session will be recorded. So I think we can slowly start and go from there with the introduction. and everything. So yeah, welcome everyone to Science Society, and of course a special warm welcome to Dr. Kim Laffey. I'm hoping I'm saying your name right.
Yep.
And oh, perfect, good. And before we start, let me introduce you to our guest speaker here today. She did her bachelor in biochemistry and developmental biology at the University of Toronto in Canada, and her PhD in biochemistry and molecular genetics at the University of Illinois at Chicago. And she was trained by Dr. Stephen Ackerman, where she studied eosinophil biology and how to antagonize them in allergic diseases. During her postdoc, she worked with Dr. Adam Schrumpf, who is the senior author of this current paper at the Mayo Clinic in Rochester. And then they moved the lab to the University of Missouri. And currently, she is an assistant research professor at the University of Missouri, and she continues working with Adam, studying the role of TCR signaling in T and B cell malignancies. Yeah, it's really an honor having you here. And if it's okay with you, before we start, we usually ask a couple of interview questions, if that's okay with you.
Yes, please go ahead.
So, how did you realize that you wanted to choose a career path in science? Was it maybe something you always wanted to do, or did it, come later, or some book that you read? If you could share that with us, thank you.
Right. Where that made me realize, oh, science is my destiny kind of thing. I think growing up, I did gravitate towards the math, the science subjects. And then biology in particular is something that I realized I wasn't bad at. So I guess I continued doing it. And then at some point, I probably reached an inflection point where it turned from something that I didn't mind to something that I quite enjoy doing. So I think it was gradual, and eventually I discovered questions that I would like to contribute solving in the field. So yeah, I pursued that in bachelor's through graduate studies, and I guess all the way till today's session.
Wonderful. I really enjoy hearing that you chose something that you actually like doing. I think it's very lucky to be able to do that. So that's wonderful to hear. And yeah, how is there maybe a story about how this specific project came about? Was it maybe really easy or hard to get funding? Was there maybe some interesting finding from a previous project that led to this, or was it like a really straight path that led to this publication?
So the first part of your question actually got cut off due to my poor signal. Were you asking what was the impetus that drove us to this particular project?
Could you rephrase that, please? You were cutting out due to signal.
Hold, let me just try to reconnect using a wireless signal and see if that is slightly better.
Yeah. Sometimes either the Wi-Fi or either the cellular data works better. I'm not sure if you're on a desktop or on the phone, but if you're on the phone, sometimes switching to cellular works better.
So how about now? Do I sound slightly more less cut out?
Yeah, now you sound good. Yep. Thank you.
Okay, let's try this connection for a while. I do apologize for this particular lack of signal in this spot of the building. Could you please repeat the last question?
Oh, yeah, sure. It was like how you came to work in this specific project, and how it came about, this project. Was it, built on something before interesting, or is there a background story? Like, was it really easy to get funding, or was it really hard? Like, yeah,
Right,
if you
yeah.
if you could share. Yeah, thank you.
So it is there is a slide of a backstory. Since my background was in allergy and the granulocytes, it might be weird that I suddenly started working on T cells, the adaptive branch of the immunity. So when I started working with Adam, I was actually thinking of continue doing things along the allergy line. And while developing that model, Adam said, we have this leukemia that nobody was looking at. I think we needed just a one last experiment, and then we can kind of see where the story is going. I was like, great, that's great time, great utilization of some downtime to do a last experiment. And of course, famous last words, one last experiment led to the next last experiment. And before we know it, it was 2 institutions 5 years later. But it was interesting that that first last experiment led to some unexpected results that we're seeing in today's paper. So it was really not a project that we expected would launch in this particular direction. And whatever I planned to do earlier, when I first joined the lab, that sort of faded into the background. So it's actually completely accidental that we started, almost accidental that we started this work.
Oh, that's really interesting. Yeah. So I'm glad you did that one last experiment. We had a guest speaker talking about deep sea brine pool discovery he did, and they were supposed to stop and go back to the surface and then find anything, and then they decided to stay 5 minutes longer, and those 5 minutes led to this humongous discovery. So it's so interesting how these things happen, and I'm really glad you you did that. So with that, the stage is yours, and please let the for everyone, the presentation is pinned on top so you can access it, and yeah, thank you.
Cool. Thank you again for the opportunity to let me share about this work with you. So I chose a journal club kind of style. So do feel free to interrupt me at any time and we can start a discussion. So today I'll just present some highlights of this research, which is really about the role of early expression and early signaling through the alpha-beta TCR in contributing to T-cell cancer generation. So throughout the presentation, I will refer to that PDF presentation, and then I will call out the slide number so that we can all follow. If you would like to refer to the original paper, it's on the next slide, on slide number 1. The link is there. So the keyword of this work is really early. What does early expression or early signaling means? So we'll spend some time defining this word early. And to do that, we need to understand some key aspects of T-cell development, which is shown on slide number 2. So the majority of the T cells that we and mice have are called conventional alpha beta T cells. They're so named because their T cell receptors have a TCR beta subunit and then a TCR alpha subunit. Now, the TCR specificities are not encoded in germline, but they have to be generated during T cell development, and that is depicted on this slide. T cells are unlike other lineages of the blood in that its development happens exclusively in the thymus and not in the bone marrow. So how this happens is that progenitors derived from the bone marrow hematopoietic stem cells will see the thymus, where they first become early thymic progenitors, or ETPs. These then go through a double-negative stage, so that is double-negative for the CD4 and CD8 surface proteins. They will then become double positive for these proteins before finally maturing into either CD4 or CD8 single positive proteins. Once they're mature, they can be exported out of the thymus into the periphery to fulfill their T cell functions. So the CD8 T cells are typically thought as cytotoxic killer cells, and then the CD4 T cells are the helper T cells. So, for example, the CD4s can help B cells make high affinity antibodies against offending pathogens. So the sequential generation of first the TCR beta subunit and then the TCR alpha subunit happens in the DN and then DP stages respectively. So let's look at that a bit in detail, and that's on slide number 3. So at the DN stage, the TCR beta subunit is first produced. And after it's produced, it has to be tested to see if it's functional. Now, this test is called the beta selection checkpoint. If the TCR beta is functional, you will be able to pair with a surrogate TCR alpha. So it's a sort of a placeholder alpha chain just to see if it's folded correctly. Now, if it is able to pair, this essentially allows it to form a pre-TCR signaling complex, and that pre-TCR allows the DN thymocyte to pass beta selection so that it can survive, proliferate, and differentiate into the next stage. The next stage is the DP stage, shown on slide 4. So at DP stage, TCR alpha is allowed to generate. And then for most thymocytes, this DP stage is the first time when the mature alpha beta TCR is first expressed. So now we can define what early expression of the alpha beta TCR means. It means earlier than DP, so it's expressed at DN stage. Slide 5. So this early expression of the alpha beta TCR is not discovered by us. In fact, it was first studied by Harold von Boehmer's group, I believe at Harvard, and Ioannis Ifantis was the first author of this JEM paper. So in this paper, they made this critical observation. That's in the mouse that is deficient for pre-TCR alpha, there are DP thymocytes. So what this means is, in the textbook T cell development model that we just covered, the DN thymocyte has to have the pre-TCR alpha, that is the gray subunit, which is a critical component of the pre-TCR. So without the pre-TCR, the DN thymocyte cannot pass beta selection and therefore cannot differentiate into DP cells. But if we look at the FACS plot, there is a great population of DP cells present in mice without the pre-TCR alpha. This means something else must be doing the job of the pre-TCR in driving proliferation and differentiation of these cells. So then on slide number 6, they found that there are DN cells that actually prematurely express the alpha beta TCR, and it's in fact the alpha beta TCR that's driving the proliferation and differentiation of cells into DP stage. And somehow that was the last people heard of these cells. There were no more studies about them until we decided to pick it back up. So just to simplify referring to these cells, we give them an acronym called EADN cells because they are early alpha beta TCR expressing cells that are double negative for CD4 and CD8. So that's really quite a mouthful. So having introduced the cell of interest to you, we can now launch into the work itself, and that's on slide number 7. So the first thing we did was to verify that we can indeed identify these EADN cells in our hands with modern flow cytometry cell markers. This is because since the last publication, a lot more cell types have been characterized in the CD4-CD8 double-negative compartment. So they are cells that may express TCR, but they do not belong to the conventional alpha-beta T-cell lineage. So we do not want to mistake those cells for our EADN cells, which can differentiate into real T cells. And this is what the slide shows. So shown here are thymocytes of a wild-type mouse thymus. So if we look into the CD4-CD8 double-negative box gate in the first panel, and then we'll remove cells that express markers for other lineages, such as gamma delta T cells, iNKT cells, or NKT cells, we can finally arrive on the fourth panel, a rare population of cells that express surface TCR beta. Now, unlike TCR beta, there is no one antibody that stains all the TCR alphas. But with whatever antibody we can get commercially, we're still able to identify cells that express surface alpha and that we have antibodies for. So showing that there are cells that express both the alpha and beta on their surface. So this will be the cells in the first box. So what this slide shows is basically we can still identify EADN cells in the mouse thymus, but they are rare. We quantify them to be right about 1% of all DN cells. So out of the thymus, they are 1% of a 1%. Now, after identifying cells, we wanted to make sure that we can see them as a phenotypically distinct subtype of cells from all these other lineages in the thymus. So on slide number 8, so on slide number 8, we did a flow cytometry experiment to make sure that they can be stably identified from other cell types. So what we did was we stained wild-type thymocytes with a bunch more markers, and then did a spade analysis. So what spade analysis is essentially a unsupervised hierarchical clustering using surface marker expression. So what we can see is that using the differential expression of this bunch of markers, double-negative thymocytes can really be clustered into distinct clusters, which can then be annotated. So the color scheme, the color scale on this particular tree graph here corresponds to surface TCR beta expression. So the warmer the color, the higher the expression. So what is immediately evident is that in our EADN cells, it has obvious expression of surface TCR beta. And this is in contrast to conventional double-negative cells, which either do not express TCR or express only the pre-TCR, which is too low to be detected by flow cytometry. There are other unconventional cells that express TCR, such as iNKT cells, but then they obviously cluster away from the EADN cells. Therefore, this shows that EADN cells can be reliably identified by flow cytometry, and they have a very distinct surface phenotype. And then on slide 9, we discover that we can see EADN cells also in human thymocytes. So shown here is a pediatric thymus. If we look at the human CD4 and CD8 double-negative cells, and similarly gate out gamma delta cells and NKT cells, we can also arrive at a population rare, but of this population that express surface TCR alpha and beta. This then is great news, because this means that if EADN cells exist both in human and mice, we can study EADN cells using mouse models. But at this point, you might ask, well, you didn't use so many markers here as you did for the mouse cells, so how can you be sure that you're looking at the same cells as the mouse thymocytes? So it is true that human DN markers are not as well worked out as in the mouse, So we got around the problem by comparing the developmental stages of the mouse versus human cells by using developmental markers. And this is shown on slide 10. So in the mouse, shown on the left, the DN stage can be further divided into substages. And mouse indel cells are at DN3 to DN4. It is not shown here, but if we stain it also with a beta selection marker, we know that they all passed beta selection. So for the T cell specialists in the audience, mouse indel cells are at DN3B to DN4. Now, we found that human indel cells actually correspond well with this mouse stage, but of course, with different markers. Human thymocytes are not divided into the DN1 to 4 system, but they are also post-beta selection. And post-beta selection is evident when the human DN thymocytes start to lose CD34, which is a stem cell marker, while with the concurrent acquisition of CD28, which is a beta selection marker. So this data shows that the human indel cells correspond well with mouse indel cells, again, giving us confidence that we can model and study indel cells using mouse models. However, remember, mouse indel cells is only 1% of all the DN thymocytes. So this still makes it a very difficult cell to study just because of how rare it is. So on slide 11, we turn to the OT1 T cell transgenic system. So in OT1 mice, all of the T cells express only one TCR, the OT1 transgenic TCR. So all their T cells will have the same specificity, and that is against some peptide sequence in chicken ova. So now, in this mice, a very important property to us is that the transgenic TCR, the OT1 TCR, is expressed early than the TCRs in wild-type thymocytes, such that a full 76% of the DN thymocytes will express the TCR, and this is much better than the 1%. But equally important to us is that the OT1 TCR is not expressed too early, but luckily for us, this TCR is actually expressed at exactly the same timing as the EADN TCR in wild-type mice. So shown on the right, it is also at DN3 to DN4. So then this is again really good for us because this shows that OT1 is a great model to study mouse EADN cells. Now on slide 12 is just a quick summary to make sure that we're all on the same page. So I told you that there exists a special type of T cell progenitors called EADN cells because they are double negative for CD4 CD8, and they have early alpha beta TCR expression. These cells exist both in the mouse and human thymus, and we can use the OT1 system to model them. And for the rest of the talk, we'll see what the OT1 system can teach us. But before that, let's actually consider just quickly the risks associated with development, and this is on slide 13. Now, the process of T cell development actually has to be tightly regulated. This is because, remember, TCRs are not encoded in the germline, that they're sequence, but they have to be generated through probabilistic genomic recombination. Therefore, quality checks must be in place to achieve an effective and non-autoimmune repertoire. But in addition, there also needs to be safeguards against the risk of transformation. And these risks can be imposed by errors during the genomic recombination or just from proliferative stress. And when such safeguards fail, transformation of thymocytes can occur in the form of TALL. Now, this T cell acute lymphoblastic leukemia is a rare but aggressive cancer of the blood. It typically involves many systems of the body, so the patients can experience problems with bone marrow function, as well as problems with the central nervous system and the respiratory systems. Regarding pathogenesis, TALL arises from transformed developing T cells. And typically, the surface phenotype of these tumors can be very heterogeneous. This is true for cancer cells within the same tumor, as well as different tumors among different patients. And this is actually easy to understand because transformation can happen to thymocytes at different developmental stages. And once a thymocyte is transformed, cancer cell, as well as its progeny, can actually still go along its original developmental program for a little bit before becoming permanently frozen in space, in time. Therefore, what you can observe is this very heterogeneous surface phenotype corresponding to thymocytes arrested at different developments. So, besides this heterogeneous surface phenotype, the molecular genetics of TALL can be equally heterogeneous. Many mutations have been characterized for this disease, but overwhelmingly, activating notch mutations is a hallmark feature of TALL. More than 60% of cases will exhibit this mutation. Now, with modern therapy, the cure rate for TALL is really not bad, at 75% for pediatric cases, but that still does speak to 25% of unmet need. And not to mention the cure rate is much lower for adult patients. There's also poor prognosis for patients with diseases that do not respond to therapy or they relapse. And lastly, TALL is less amenable to CAR T cell therapy, which has been successful to treat other blood cancers. And this is because this is a T cell cancer. So firstly, it is actually quite difficult to harvest enough healthy T cells from the patient to engineer CAR Ts out of. Secondly, if the CAR Ts are engineered to kill T cells, they will probably kill themselves. So antigen selection can be a challenge. Therefore, to address this unmet need, I think there is great urgency in understanding the pathogenesis of the disease better. But there's also urgency in integrating immunophenotype with the underlying mechanism of transformation so that we can better stratify the risk of the patients as well as discover maybe new avenues for intervention. And it is in this spirit that we undertook this project. So slide 14. First, we realized EADN cells in humans can transform, and transformed EADN cells can form part of a TALL tumor. And this is illustrated by patient number 1. So there's a lot going on this slide, but I will take you through it. So in panel A, we are seeing is the peripheral blood of the patient. What we can see is that there are actually residual healthy T cells in this patient, such as the single positive CD8 cells. So if we quickly look into panel B, we can see that the CD8 single positive cells are T cells because they express the CD7 T cell marker, and they are positive for surface TCR as expected, and this is marked by surface CD3 expression. But other than these residual healthy T cells, there is a huge smear of cells expressing variable CD4, CD8, and this is the bulk tumor. So what about the double-negative cells of this patient? If we look at the DN cells, we can indeed also see a small population of T cells because they are CD7 positive, but they also express surface TCR. So they are starting to satisfy our criteria for being an EADN cell, in that they are DN, they have TCR. But how do we know that they are transformed EADN cells? Well, we can look at their CD45 expression in panel C. So in human T cell development, CD45 level tracks with maturity. So relative to the mature, healthy CD8 single positive cells represented by the gray histogram, we can see that the transformed EADN cells have less CD45. Now, because we do not express, we do not expect healthy immature thymocytes in the periphery, so these immature cells must be the blastic malignant cells. So because in this patient, all T cells are also EADN cells, we can say that EADN stage is the phenotypically earlier stage that we can identify in this TIL case. But this is not the case for the second patient. If we look at the blood of the second patient in the lower panels, this patient actually looks broadly similar to the first in that we can also see residual healthy T cells, but also this big smear of CD4, CD8 heterogeneous population representing the bulk tumor. If we look into the DNA compartment of this patient, we can actually see that TCR expression is variable for CD7 positive T cells. Most of the T cells will be negative for TCR, but some also has deemed TCR. So how this patient is different than the first is that transformation probably happened in a cell that is earlier than EADN stage, when TCR expression has not happened yet. And this last point can really be better seen if we look at the bone marrow biopsies of these patients, and that is shown on the next slide, slide 15. So if we look to the right, what we can see for the first patient is that the bone marrow is completely infiltrated by cancer cells, and all the cancer cells express the alpha-beta TCR, as indicated by the brown staining. In contrast, the cancer cells do not express gamma-delta TCR, which is the alternate T cell lineage. So at this point, I would like to bring up just another aspect of T cell development. So when a thymocyte passes when a thymocyte passes beta selection, it actually commits to the alpha-beta lineage, such that it loses the ability to pursue the alternate gamma-delta lineage. So that this data is very much in agreement with EADN cell being a post-beta selection cell type. Now contrast this with the second patient. Right, the second patient's bone marrow is also heavily infiltrated by tumor cells, but it is infiltrated by tumor cells that express the alpha-beta TCR and is infiltrated by cancer cells that express the gamma-delta TCR. So again, this shows that transformation happened in a cell that still retained or that still had the ability to pursue either the alpha or the alpha-beta or the gamma-delta lineage, so it happened earlier. So what this collectively data is showing is that EADN cells can transform, and we can identify EADN stage transformation from other stage transformation.
Do you take a quick question clarification?
Yes.
It's in the room chat. Shall I read it for you, or could you read it yourself? If you slide from left to right.
It might be
I
easier
think,
if you read it for me.
yeah, I could just read it. So the question is, do the cure rates at 75% in pediatrics and 50% in adults include 5 years free of T-all malignancy, or only current cure rate of eradication of malignancy in oncology patients presenting with T-all? That is a question by Christy Berkeley.
Thank you for the question. So I'm not a clinician, but I do believe the cure rate represents, I want to say, response to first induction therapy. Now, the relapse, I believe, well, the relapse is about 25% in pediatric patients. I can't say for sure, just because I'm not a clinician.
Christy, could you clarify your question a bit?
I was just wondering, well, when I saw the figures, that I was just wondering about if the cure rates that you had mentioned in your slide, because I've been following along with your slides, if they were just for the patients as they presented. I just wondered if it included like a 5-year free of cancer. But it is, so you're saying the cure rate that you have on your slide is just for the initial presentation of TALL.
Right.
Is that right?
I think so. I think it's mostly for the initial presentation. But if they do relapse, the cure rate is very bleak at that point. So even if I underestimated, you will not add much more to that number, I think.
Okay.
If
No,
that makes sense.
I was just curious because I just was wondering if it was just for initial presentation or if that included a 5-year cancer-free rate. So thank you for answering my question. I appreciate it. And I'm enjoying your presentation.
Thank you. Thank you. Okay. So we can continue. So for the human data, I'll just restate the conclusion that EADN cells can transform, and this particular stage of transformation is distinct from if transformation has happened during other stages. Now, on slide 16, very serendipitously for us, we observed that OT1 mice can spontaneously undergo EADN stage transformation. This basically makes OT1 an even greater model because now not only can we study healthy EADN cells in the OT1 system, we can also study transformed EADN cells in the OT1 system. And we found that the TALLs in these OT1 mice recapitulate the human disease quite well. So if we look at the gross presentation in figure A, TALL in OT1 mice is abbreviated TOT. So if we look at the top picture, we can see the lymphoid organs are hugely enlarged with cancer cells. So first shown is a large spleen, and then thymus, and then various lymph nodes. So this again is quite in agreement with the human clinical presentation of the disease. And if we look at the surface phenotype of the tumors, we can also see in panels F, G, and H this characteristic smear of CD4 and CD8 expression, also reminiscent of the human disease. Now, how do we know that this is EADN stage transformation? Well, if we look into panel D, both in healthy and leukemic mice, all of the T cells basically express surface OT1 TCR. So that means any double-negative cells in the tumor will also be TCR positive. So if they're DN, they're TCR positive, they satisfy the criteria for being an EADN cell. So this shows that OT1 mice can spontaneously undergo EADN stage transformation. Another point that proves that we have a good disease model in our hands is on slide 17. So remember I said activating notch mutations is a cardinal feature of TALL. So it is gratifying to see that OT1 TALs in this mouse model also harbor activating notch mutations. So what this table shows is that for all these primary tumors we sequenced, each tumor has a unique mutation. So this unique mutation also establishes that each tumor is clonal. Therefore, again, showing that the OT1 system is able to spontaneously generate TALs that's reminiscent of the human disease. Now, at this point, we were really curious as to what is causing the TALs in this system. And our first clue that TCR signaling might be involved came from this cohort study shown on next slide, on slide 18. A very little bit of background is that in mature peripheral T cell signaling, 3 things are required. The TCR, of course, the MHC presenting antigen to the TCR. This is because TCRs cannot recognize just free-floating antigen. It has to be presented to it by MHC expressed on antigen-presenting cells. And the third thing is a co-receptor, either CD4 or CD8, but that is not depicted in this cartoon. So the 3 components of TCR signaling, and that is all you need to understand the remainder of the slides. So in this experiment, we followed different types of mice. to see which mice will develop TALL over some observation period. At first, we can see is that wild-type mice do not develop TAL. But in OT1 mice, as I've showed you, do develop TAL, and the incidence is about 10%. However, if we introduce the beta-2M mutation, which abolishes MHC, so abolishes TCR signaling, the tumor goes away. Now, of course, it is very difficult to prove zero, but we tried our best by following 190 mice up to a year, and we still see no tumors. Therefore, we are quite confident in saying that for this model, TCR signaling is required for TALL generation. We also looked into some other mice to try to discern other rules of transformation. Now, the REG mutation removes the ability of the cell to cut DNA and therefore to generate TCRs or even B-cell receptors. So the fact that a REG knockout is able to also develop TALL shows that REG-mediated DNA recombination is not required for TAL generation in this model. The remaining two types of mice are really just a control type of control mice to show that cancer generation in our hands is not due to some special mutation that's specific for our colony of mice. After establishing that TCR signaling is required for tumor generation, we next also wanted to find out if the continued growth of the tumor cells still require TCR signaling. So we did this experiment on slide 19. So what we did here was that we injected early TAL cells into wild-type mice that is capable of TCR signaling, as well as MHC deficient mice that are incapable of TCR signaling. Early TAL cells means that these tumors have not been passaged through mice many times. So they haven't had a lot of time to evolve away from the primary tumor. So as we can see from the photos, the tumor cells had no problem growing in wild-type mice, which is capable of TCR signaling. So we can see this characteristic enlargement of the lymphoid organs, and the size of the spleen is quantified in the graph. What we can also see in the graph is that in beta-2M knockout mice, so mice that are incapable of TCR signaling, tumors cannot grow. So this data shows that the continued growth of the tumor cells is also dependent on TCR signaling. But what happens if we now allow the tumor cells to evolve? Let's see on slide 20. So here we serially passaged tumor cells through mice many times. So as expected, the tumor cells became more and more aggressive, and the mice succumbed faster and faster. In fact, the tumor cells can become so aggressive that we can derive a line to grow in the dish, and these are the TALT1 SI cells. So we injected these very aggressive cells into mice that are not capable of TCR signaling. But if the cells can grow in the dish, they will have no problem growing in a mouse without MHC. So indeed, this is what we see as quantified in the graph, last column of the graph. These late evolved aggressive tumor cells are indeed able to grow in mice without MHC. So this shows that with tumor evolution, MHC requirement can be overcome. So just to summarize what I told you so far about this oncogenesis model, and this is on slide 21, is that TCR signaling somehow can give rise to transformed immune cells bearing activating notch mutations. And if TCR signaling is allowed to happen, this can eventually lead to continued growth of the tumor, and this leads to overt cancer. But with tumor evolution, the tumors can gain aggression such that the MHC requirement for growth is lost. The empty space on the right is intentional. because we wanted to see what was happening to the yet healthy and untransformed Eden cells that kicked off these downstream events. And this is what we did on slide 22 to try to figure that out. So now we're back to untransformed healthy Eden cells. So in mature peripheral T cells, TCR signaling requires co-receptor, either CD4 or CD8. But we find that this is not the case for immature Eden thymocytes. So when we looked at NER77 expression, which is a marker for TCR activation in mice that are deficient for all types of MHC, so they are not capable of TCR signaling, versus wild-type mice that can do TCR signaling. We see that Eden cells in wild-type B6 mice express higher NER77. So this indicates that Eden cells can respond to endogenous MHC ligands in a co-receptor independent way. Now, this observation can be recapitulated also in the OT1 system in a more controlled fashion, and this is shown in panel D. So in this experiment, OT1 thymus is isolated from mice without MHC, so that these OT1 Eden cells would have never been exposed to endogenous TCR signaling. And then we can culture them with exogenously added MHC presenting different peptides for the OT1 TCR. So what we can see in the graph is relative to the negative control peptide FARL, OT1 Eden cells is able to upregulate the activation marker CD69 in response to its strong antigen over peptide. So now, because we know Eden cells are negative for the co-receptors, any response to the signaling peptides must be co-receptor independent. Now, interestingly, this property to signal without co-receptors is also retained in transformed Eden cells. And this is shown on slide 23. So on this slide, we did sort of a similar experiment just with the transformed Eden cells that you've seen before. We use these cells because they have a classic Eden phenotype. So if we look at the flow plots in A, the left two panels show that these transformed Eden cells are double negative for CD4, CD8, and they have surface TCR expression. The right two panels show that they are a post-beta selection cell type. So they are at the expected developmental stage that we expect Eden cells to be. So then in B, we again culture these cells with antigen-presenting cells presenting the various peptides for the OT1 TCR. So very similar to what we saw in the healthy Eden cells, relative to the negative control FAR peptide, the transformed Eden cells is able to upregulate activation marker in response to the weak Q4H7 peptide as well as the strong overpeptide. And this response is not impacted at all when we block CD8, which is the co-receptor used by OT1. So again, this shows that in both healthy and transformed Eden cells, they are capable of TCR signaling without co-receptor. And this is very different than what we understand of TCR signaling just in conventional T cells. So to summarize what I told you and to put everything in kind of the context of the entire story, on slide 24 is this multi-hit model for Eden TAL generation. So in this model, Eden cells, healthy Eden cells with wild-type Notch protein is capable of co-receptor independent signaling. Now, through a yet unknown number of other steps, somehow it can lead to the generation of transformed Eden cells with activating Notch mutations. And with continued TCR signaling, this can lead to overt cancer that can be very aggressive. So co-receptor independent signaling in this model is really the driver event for this TAL generation. Now, it's important to note that there can be many mechanisms to lead to TALL, and we're only characterizing this particular mechanism in our work. So still many questions remain, and this project is actively ongoing. Some of the things that we are currently looking at is presented on slide 25. So on the developmental side, we would like to further characterize the cellular landscape of the EADN cells using omics approaches. So this means looking at their genes, looking at their signaling pathways, and we hope to find a clue as to why this particular stage is susceptible to oncogenic transformation. Now, on the cancer side, we certainly want to find out what is the prevalence among TALs that actually exhibit this EADN stage transformation. And does this stage transformation have any impact on how the disease responds or relapse in response to treatment? So we hope findings from this endeavor will help us improve risk stratification. And finally, just the acknowledgement slide. This work really benefited from the generous help of many people over many years across 2 institutions. Many of these people are my co-authors, but today I would just like to, in particular, acknowledge my PI, Dr. Adam Schramm, because he is ultimately responsible for creating a very pleasant, very collaborative, and highly effective research team out of this many of us. And thank you for your attention, and I'll be happy to hear your thoughts.
Yeah, thank you so much, Kim, for this wonderful presentation and for explaining this to us in such a way that, you know, I could understand it. So I think everyone then should do it. So it's so interesting, the work you did, and it will, I think, help a lot of people understand these mechanisms so much better. So congratulations again for your work, and for it must have been very labor intensive. So how long, just that people get a better idea, how long did it take to do all this research that you presented today?
Oh, so the project itself lasted, I believe, 12 years. Because from the initial identification of the tumors to the actual elucidation of the mechanism, that was a very long time. But I guess the mechanism is what people are most interested in, and that's the most of the part where I came in. And this was done in the last 5 years. So it is a long journey, I think.
Yeah, wow, that's that's really good, you know, to point that out. I think you know, it gets condensed into these images and into these papers, but I think it's really you know, important to point out like how much amazing work this was, and to get to this knowledge, and so yeah, so thank you for that. And so you it was mentioned before, and also an article that I read about your paper, that an autoimmune reaction can cause this EADN to turn into cancer, and that, you know, many cells in the thymus can not do this. So this autoimmune reaction, do it's, do you know, I know it's not like very specific to this field, but for people to know, like, what can trigger this? Is there something that can trigger this specifically? You know, just for people maybe to get an idea how you know how these get triggered, or if it's just something that just happens.
Right. So the short answer is we don't know. But the word autoimmune really just means reacting to self. And the ability to react to self is a critical part of how T cells develop. So to be a functional T cell that leaves the thymus and be able to function in the body, it really has to go through some kind of calibration. So first, it must be able to react to self, actually, for a little bit, just to make sure that the TCRs are functional, are working as they should. So they need a little bit of reaction. And this just confirms that, yep, you are made correctly, so you can go on. But if the next step, the T cell is found to react too much to the self, then they will die. Because then if this T cell is allowed to leave the thymus, it will then create really autoimmune diseases or reactions that will be harmful to the host. So for cancer generation in this particular model, what we currently cannot distinguish will be two possibilities. One is there's nothing wrong with the body itself. So what the developing T cell sees is what every other healthy developing T cell sees, right? There's nothing inherently wrong with the structure, with the rest of the body. But that T cell, that thymocyte itself, might have a failure in calibration of sensitivity. So you might be seeing an antigen that is supposed to tickle itself a little bit, but it has now an over-exuberant response, and you might go cancer. So that is one possibility. The other possibility is maybe something is wrong with the rest of the body. Maybe the T cell itself is perfectly okay, but maybe it's seeing a particular antigen, and that antigen is triggering a stronger than normal response. And that causes the thymocyte to, again, mount an over-exuberant response and go cancer. So currently, we are trying to figure that out. Because the TCR is involved, it certainly involves antigen and receptor, but we just don't know which side the problem lies. It could be both.
So that's really interesting. Yeah, it could be both, and maybe it is. So do you think that will be a way to pre-screen the risk, you know, this ongoing research?
Yeah, that is. So in general, TALL is extremely rare. So just on the population level, it seems that people have decided that there's not much benefit in screening for this disease. I believe it's in the single digits per million children. So it is an exceedingly rare disease. And among all those cases, to have this particular subtype of TAL, I think it will be even rarer. So while it will be great if there is a way to successfully screen for it, I wonder if the barrier is more with the efficiency of the exercise.
But do you think that knowing this mechanism could could be a mechanism that could, like a principle that could be important also for other cancer triggering mechanisms? Basically, if if you find out what the predisposition basically is.
Yeah, that is a very interesting point. That is a very good point. That really deserves some thought, because the ability of the developing thymocyte to react and to mount an inappropriate response and is allowed to get out of the thymus will have huge implications. So I think you make a very good point in not only cancer development, but the development of autoimmune diseases. That is a good point. Yes, thank you.
So related both to autoimmune diseases and potentially finding subsets of the population where it is not single digits per million. When you mentioned that these cells can be MHC independent, I immediately thought of the question, or that immediately raised the question, can it be MHC dependent but antigen dependent? And refining the question further, are there important special cases where you have antigens that are not perfectly bound by MHC? So this is like when, if you think about, say, a response to a virus where you have a T cell epitope and there's a mutation in that epitope, so that it doesn't bind perfectly, and the result of that is something like either autoimmunity or on the other side, T cell anergy or apoptosis.
Right. Yeah, I think that is a good point. In this case, the host has nothing wrong, right? The T cell development is going on correctly, and the antigen presentation, the selection system in the thymus is functioning correctly, but you will have some sort of infection, and that exogenous antigen is causing is causing an aberrant reaction, and that results in a bad clone escaping, either as a tumor clone or an autoimmune clone. Yes, that I believe that is an implication that we should think about.
Sounds very interesting. Thanks.
Thank you.
I wonder what the effect of an infection would be on this process, or a chronic infection.
That is a good point, and this is probably a future aspect that we should think about, because in mouse models, these mice are kept specifically away from sources of infection. But I think that definitely is something that we want to look into, particularly because we have the intention of looking at what kind of TCRs, if there's a bias in some types of TCRs that are more oncogenic. So an implication of that will be, are antigens then also biased? If some antigens are, that there's a preference for some antigens, then that will speak to the persistence of low-level but chronic antigens, such as from a chronic infection. So yes, those are definitely important areas that we still need to unpick in this model.
Thank you for your answer.
Thank you for your question.
I wonder if Katerina has unmiked or not. I don't hear her.
Can you hear me
So
now? I'm
Yeah.
sorry. Oh, I'm sorry. Somehow the unmiking didn't work before. So yeah, if anyone has any questions, please go ahead. Denise, LT, Swampy here, I saw you joined. Please go ahead.
I do have a question, but Denise, do you have one ready? You can go ahead, please.
After you.
All right. Thank you. The great presentation. Thank you. I have a question is related about when you said that about 60% of the tumors you follow up, they have this notch mutation. If I remember correctly, notch has a family, right? Is it which notch molecule will associated with this particular kind of line of research? Thank you.
this is notch 1.
Notch 1. Okay. Oh, okay.
Yeah.
I missed that. And then is it possible to sort of like have people, if people, I was thinking of early detection. Is that maybe too low a percentage to thinking even in that line of thought? Thank you.
Do you mean early detection in people
Yeah, might be. Yeah, people
who
like
have
because
not, just apparently healthy people?
yes,
Ah,
you got me. Yeah.
right. So right now, just overall, I think epidemiologists do not recommend screening for TALL just because this whole disease is very rare. So they feel, maybe the efficiency of this whole undertaking is not worth the benefit of catching the disease because just it's so rare. You're most likely to get
Other
no
kind, I said
patients.
before you, I
Right.
see, gotcha. So very real, meaning how real? Probably I missed the beginning of your talk. Did you say
No problem.
something in this?
Right. I actually don't think I went through that. I believe
Okay.
it's 3 or 4 new cases per 1
Oh, okay.
million. Yeah.
1 minute. Okay.
Yeah.
Thank you so much. Yeah, then I should not be thinking in this way. Thank you. No more
Yeah.
questions
No
for now.
problem. Thank you.
Thank you so much for your amazing presentation. I had a very basic question, which is, how does one test for TAL? Is it a very difficult test to run? Is it obviously once it scales up, it can be inexpensive, but it's not. Doesn't seem like it's necessary from an epidemiology standpoint, or what would it take to test for a TAL? How long? How much?
Right. So there is really no at-home test. What people, how people are seeing currently is really patients start having, the very overt signs of being ill. So again, I'm not a physician, so I will not know the specific symptoms, but I do believe there will be night sweats, fever, maybe difficulty in breathing, depending on what the cancer is, what system the cancer is affecting. Only when it's very severe, the patient is presented. The patient presents at the hospital. And that's when they will do a biopsy and find cancer cells in the blood. So I believe that this is currently the way that TALLs are discovered. I don't know if just a regular blood work without looking for specific markers, people routinely look for this as a possibility, just again because this is so rare. It's only when maybe you have an extremely high white count, and then they might start suspecting, and I wonder, that then it has to be the age group and everything, and then by probably they will eventually come to the conclusion that TAL is a likely cause. So again, I don't think this is can be an over-the-counter kind of test.
Sure, I understand that. I mean, just in the context of the work that you're doing, how long does it take to test a specimen, for example? And is that where is that just blood work or?
So if then it will be blood work. If you want to confirm if the patient is TALL and what type of TALL it is, then you will be blood work or maybe bone marrow biopsy to stain for T cell markers. If they are immature looking T cells in the peripheral blood, typically that's a very good sign that it's a TALL. Because there really shouldn't be immature T cells in the periphery, and then I want to say that a typical practice now is to also send the bone marrow or I want to say bone marrow for sequencing. So you will actually check for common mutations such as notch mutations or MHC mutations. So there's, I guess, a standard panel both in the cellular marker expression as well as the a mutation panel. And then that will be confirmed as TAL in diagnosis.
Understood. Thank you so much.
No
Who else has questions? Katerina, perhaps you have another question?
Yeah, can you hear me? Perfect. Swampy, I know you joined the stage. Do you want to add a comment or ask something, or did you just want to join us on the stage?
I was kind of reading a little bit on this, by the way, on the internet, and it sounds well advanced beyond my protocol. I was going to ask you the thing about the thymus. My understanding from leukemia is, that it gets in the bone marrow, and I see this, and I know this drilling down as far as the cells and what works and what does not. The thymus, is that part of the thyroid? I know a lot of times when people go to get their blood work done, okay, they check about their thyroid. Can you tell me about the relationship between this rare, I guess is it like a virus, and the leukemia and in the bone marrow, okay, and the thyroid? Is there any correlation there, or I'm just taking like thyroid and making it an inferment thymus? Thank you.
The thyroid and the thymus are completely different organs. I would not know anything about the thyroid simply because that is not my area. So the thymus is part of the immune system. It is right above the heart, actually. So I would say, without MD's knowledge, it is relatively close to the thyroid, I would say. But the thymus is really important when we are young. When we reach puberty, the thymus kind of just shrivels up, and it no longer is very important in pumping out new T cells. We establish our our repertoire of T cells before we reach puberty. So the thymus is the site of T cell development and maturation, and it is the organ where T cells know what not to attack itself, but to attack everything else. So not to attack the human itself, but attack everything else that it sees. So virus, yes. So typically, it will not go into the bone marrow. If you see T cell-like stuff in the bone marrow, that typically is a sign of cancer. And how cancer gets into the bone marrow is probably through, abnormal receptor expressions, and they sense other cellular signals that kind of draw them to the bone marrow. But yeah, the thymus is the primary site for T cell development.
Yeah, if I may add, there's a really cool, I don't know if anyone knows NPR Radiolab. There is a really cool Radiolab podcast session about the thymus, where it describes really well, how the thymus is basically the training center of the immune cells. So the thymus has like a tiny copy of you and all your different cell types, basically, or cell surfaces, and then the immune cells get trained in the thymus, and I think something like 80% don't make it through the training. It's as wasteful as the brain development, I think, or about similar. So if they keep not recognizing what's you and what isn't you, the cells get destroyed. So, but it's kind of, you can imagine like a training center for your immune cells, and the thymus is like a mini you. So that it's really interesting. And there's a researcher, she is trying to take a thymus from donors and grow them like little slices and grow them in the petri dish, and then expose them basically to the immune cells of the recipient, and kind of apparently the thymus is able to learn the recipient's um, environment, so like to copy when you transplant it, to copy basically the mini you of the recipient, and to treat like leukemia and stuff in kids. It's really interesting, so I just recommend that podcast. Sorry, Kim, go ahead.
No, that is an excellent description and summary. That I that couldn't have done better. That is actually, I want to listen to that podcast too.
Yeah, it's really amazing. So yeah, I can only recommend it. Thank you. Yeah, we've been going.
Thanks.
Oh yeah.
I
One
just
last
wanted,
thing. Go ahead. I'm
sorry,
sorry.
go ahead. I just wanted to thank Dr. I'm sorry, Dr. Laffey for the presentation. It was incredibly interesting to listen to, and I appreciate the slideshow that you went over with your research. And I guess I just had one question: is the next step in the research that you're doing is to determine, are you going to try to determine how frequently in humans all TALL cases originate from these Eden cells, or is that not the direction you'd be going in?
Yeah, so that is certainly something that we'd like to do. So we want to see just how many percent of TALLs exhibit this Eden transformation characteristics, and see if that impacts how the disease responds to treatment, or whether these diseases are more or less likely to relapse. Because right now, how doctors classify TALLs is really by looking at the bulk of the tumor. It is all double negative, and this is one bucket, or it's all double negative, it is one bucket. But we are almost looking at the minor population that is present in the tumor. So we want to see if there's any value in doing this. It could be that there's no value, there's no correlation whatsoever, but that's not typically what's done. So we really wanted to see if this minor population that's present in the TALL bulk, are they the cells that are responsible for relapse, or are they the cells that won't get killed by chemotherapy? So that is certainly what we're trying to do next.
Swampy, did you have a follow-up comment or question?
I did, and again, I apologize, Dr. Laffey. I got in a little bit late and kind of a little bit learning on this. The
No
again thing
problem.
about the thymus you'd mentioned as far as the immune system, is this really being looked at closely with regard to COVID? I know a lot from what I've heard. The COVID works inside the lungs, and it has, I guess, an antibody or a reactant, right, that makes it accumulate lots of fluid. And again, I apologize for not using the medical terminology. And basically, the person has pneumonia. I was in an automobile accident with a 41-ton rock truck, and I got what's called mechanical pneumonia, in which my lungs were filling up with fluid, and I had to take some blue crystals. But the connection with is the thymus. How important is that with regard to the immune system? Does it contribute, or is it the main source for immune system? And if so, has there been a lot of research on that with respect to the COVID? Thank you.
So with regard to COVID, the T cells aspect of it is probably the slightly less characterized arm of COVID response, because what we've been focusing is the antibodies production, which are produced by B cells. That's because it's actually a lot easier to measure B cells in the blood, because to measure T cells and COVID-specific T cells, that requires a lot more doing. So it is not easily done. It cannot just be draw some blood and test. So, but the adaptive immunity, T cells and B cells are equally important, and in fact, B cells cannot produce high affinity antibodies without help of without help of T cells. So we do believe that T cells form a very critical component of the anti-COVID response, and they are, I guess, there are conjectures or they are ongoing research into probably T cells will be forming the much more lasting immunity against COVID, even as the B cell antibodies start to go down. But again, it's just not easy to measure COVID-specific T cells. Therefore, a lot less is known about the T cell branch versus the B cell branch. As to regard to how important T cell, the thymus T cell and response to diseases in general, and I think because the thymus, the T cell repertoire is really established early in our lifetime. So the aging of the repertoire is important. Like I said, after puberty, our thymus really become fibrous, involute. They don't function as much anymore. So whatever T cells that we're armed with, we probably will depend on them for the rest of our lives. So And I believe that is one contributing factor that age might be sort of a predictor for a bad COVID outcome. Part of it is the number of available types of T cells that can respond to COVID decreases as we age, right? We have so many good T cells, but as we age, like everything wears out, things start failing, things start dying. So we kind of just lose some weapons in our weaponry, if that's an analogy. So age, the thymus no longer putting out as many new T cells as we age, all that has an impact on sort of a complex response to COVID as we see over a population level.
Thank you very much. And then as far as the DNA, okay, my understanding, that's the reason that we age, because there's perhaps a mutation in the genes. And I think overall that might be somewhat similar. As far as the thymus, and it's basically degenerative over age. Is there any last thing is I was going to ask you? Is there any healthy ways to encourage or increase the operability of the thymus, or is it unable to be sustained? For example, like the liver, not drinking as much alcohol, not eating processed foods. Because seriously, this is the first time I've ever heard of the thymus. Thank you, Dr. Laffey. And I'm just kind of curious: is there anything that can help delay, okay, its degenerative operability?
So there's, for the most part, there's no way to reverse the evolution of the thymus. It is part of a physiology. And for the most part, we don't have to, because we all kind of live pretty happily, even though by now, since most of us should be post-puberty, all of the thymus would have evoluted. So there really is not much added bonus to the boost in immunity, to rejuvenate the thymus. But of course, there are specific conditions where medical conditions where if they had a better thymus output, you probably will boost their immunity. But that's to boost it to a normal level, not boost to, I don't know, a superhuman level. I don't think that's needed. As to foods and all that, I honestly don't know. I would say just exercise more, quit smoking.
Yeah, the late Dr. Wayne Dyer, he had leukemia, and supposedly there was a diet called the APO diet, okay, and I think it delayed that. It gave him several more years. Not really sure about that, because I'm big on science versus wellness, natural in general, because I know the science, you all have to go through a lot in order for it to be passed using the scientific method. But yeah, leukemia, thank you for sharing that this is a rare form of that, okay? But yeah, the leukemia, as far as I heard getting into the lymph glands, like cancer in general, very hard to treat if you catch it late once it gets into the lymph glands. And that's all. Thank you very much.
Yeah, thank
Yep,
you so much.
thank
I
you.
wanted to add that there was a paper that came out earlier this year that if children grow up under high stress circumstances, their immune system stops maturing prematurely, so they kind of, I think, it stops too early, so they don't have a chance to learn as much how to. That's the only study I know that looks like it in environmental factors. But I saw Eli and Mike. Please go ahead.
Yeah, before I ask what I was going to, or mention what I was going to mention, Katerina, when you say stop developing early, does that mean increased autoimmunity, or how does that manifest?
Yeah, so they didn't know that yet. They just said that it matures too early, so it kind of stops the development earlier than in, in control group where they don't grow up in high stress levels. That's what I know right now, but I will check into the paper again and then just reach out to me. I can share it with you.
Okay, thanks. Yeah, so there was the mention of thymus rejuvenation, and of course that's a really speculative topic, but it has been coming up in connection with what is being seen in people who have had COVID infections, because one of the things that seems to be happening is depletion of naive T cells, although it isn't unambiguous that this is just depletion of circulating naive T cells, as opposed to what's also going on in the bone marrow. But the argument is being made that there is a restriction of through potentially a number of mechanisms, restriction of the naive repertoire by people who've undergone COVID infection, and especially people who have experienced long COVID. And so I'm wondering if you have any thoughts on that.
I'm actually not very familiar with that branch of research. So I'm not sure if with COVID there is suppressed, so you're saying there is both suppressed bone marrow output in addition to the wiping out of peripheral naive repertoire?
Well, so it's circulating naive T cells that seem to be depleted, and the question is this reflective of the reserve, so to speak, in the bone marrow? But this has been raised as a potential reason that people might become more susceptible to infections in general, new infections.
This, I really don't know how to speculate on that, because I'm simply not, I'm not able to connect bone marrow output and then to thymic output, because that will also be affected by the age of the thymus.
Yeah, I believe this is in adults, right? So we're
Right,
talking about
in
post.
adults. Yeah, I wouldn't know. Also because it apparently does not happen to other people. I mean, it does not happen to everyone. So then, right, if it happens to everyone, we can maybe easier to derive some clues as to what is impacting the journey from, say, a common lymphoid progenitor getting to the thymus and then finally maturing. But if there's so much variability in COVID response, and even in people of the same age group, I think it's, well, human are not mice, so the genetics and the human-to-human variability is so much. It is hard for me to speculate.
No, absolutely fair. I mean, COVID's kind of a mess, so not surprising.
Yeah, thank you so much. We've been going like for over an hour and a half, so just thank you so much for sharing your work and the presentation. And it's really interesting, and I'm really curious to see what your next results are based on what you shared with us here today. So I really appreciate it, and thank you everyone for coming and asking questions. Sorry, my kids.
Yeah, thank you so much.
And yeah, we wish you all the best for your research, and a lot of funding, and yeah, I hope we'll
Yes.
get to learn some more from you one day. Thank you.
Thank you again for the invitation, and this was really fun for me too. Thank you.
Yeah, I'm glad to hear that you enjoyed it too. That's very important. So yeah, thank you. Have a wonderful long weekend. Enjoy your long weekend off, and yeah, I hope I hear you all back soon again. We will have more rooms next week, so follow the club if you like discussions like this. We have on Monday Dr. Ruprecht from Germany coming. He will talk about this astrocyte work and how he established them as computational units. Then we'll have on Tuesday glioblastoma and how the brain invasion works on and yeah, for cancer. It's cancer research. And then we'll have a Di Pascali from Italy joining us to talk about his 3D printed biomimetic artificial muscles that he developed. And then we'll have mutations that protect from cognitive decline. And Thursday we'll have my friend and previous alumni. He has research about goal-directed and habitual actions. That's really interesting research in that field. And on Friday, we'll have Dr. Michael Levine from Tufts. If you heard about the xenobots research that was kind of in the news, he did that, and he was here before and shared his research, and he will share his current ongoing lab results with us that are also unpublished. So if you want to get a peek preview into what's going to be published out of his lab, join us next Friday. Okay, thank you again, Kim. Enjoy your weekend, and hopefully I hear you all next soon.
Thank you so much. You too.
Close the room in 3, 2, 1. Bye, everyone.